Good morning, everyone. Welcome to week 4 of your journey into the world of the unseen. So, this is week 4. This is the middle of the term, which means that you have your midterm exam to consider this week. More on that in a little bit. So, I have posted links to two videos to help you understand the concepts that you'll be looking at here in week 4. The first describes fungi and protozoans, and in the second, Professor Dave describes viruses and explains how viruses like SARS-CoV-2, the causative agent of COVID-19, are able to get into cells and essentially hijack cellular mechanisms to make you sick. So we'll touch on two topics this week. In chapter 12, you'll be looking at eukaryotic microorganisms. Not all microorganisms are bacteria. Some of them are eukaryotic, meaning their cells have a true nucleus. Things like amoeba that you can pick up in contaminated water can cause all sorts of um, problems with your digestion, for example. So when people think of microbiology, microorganisms, they generally picture in their minds a bacteria. But there are lots of eukaryotic microorganisms and pathogenic microorganisms. Um, so organisms like fungi and protozoans, I mentioned amoeba, even multicellular organisms like tapeworms. Interestingly, even though you can see tapeworms and roundworms and pinworms with the naked eye, with the unaided eye, for some reason in history, those organisms got grouped in with microscopic pathogens. And so we still talk about them today as part of microbiology, even though, again, they are not microscopic. Okay, that's chapter 12. And then chapter 13, you'll look at viruses, virions, and prions. It's a misnomer to call viruses and prions living. They're not living organisms. They are organic nonetheless. And so something like um, Ebola or SARS-CoV-2 are tiny packets of nucleic acid. So they have DNA and they have RNA wrapped in a protein package. And then they attach themselves to bacteria, they could. Those are your bacteriophages, or eukaryotic cells like your body cells, and then introduce their, essentially inject their um, genome into your cells, and then your cells recognizes the genome as DNA or RNA, incorporates it into your cell's own genome, and then your protein, your translation mechanisms um, translate the viral or the prion um, genetic material into proteins that then destroy your cells and make you sick. Viruses are extremely fragile on the outside. They cannot do anything. They are obligate intracellular parasites, which means they have to invade your cells in order to do anything. And remember, they don't really do anything on their own, like bacteria do stuff on their own, but viruses don't do anything on their own. They have to infect your cells or a cell in order to be able to effect have their effect. They're unlike bacteria, which their job as a biologic organism, as I've explained before, is to reproduce and propagate the species. Viruses are not like that. They don't have a quote unquote, I use this term very lightly, they don't have a brain as such, like a eukaryotic cell or a bacterium that tells them to propagate and reproduce. they just do it. Um, so that's something to remember for the future. There's no protein mechanism. We say they're self they're self they self assembly or self assemble inside of your cells or inside of a bacterial cell without any prompting from any kind of protein mechanism. All right. Okay. Now viruses are too small to be seen with standard light microscopes, such that we use in 200 level microbiology courses. Simple compound light microscope. We can see bacteria for sure. Cannot see viruses. They must be viewed with the very sophisticated and expensive electron microscope. Okay, housekeeping items for the week. Make your discussion post early in the week so you can fully engage in the discussion. Now, your topic 4 post to do no later than this Friday. It's January 30, 11:59 p.m. GCU time. I want to say this about discussion. Some of you are doing really a fantastic job on discussions. Please, you need to go to class resources and review my guidelines for discussion posts because some of you are not making the 150 minimum word substantive length. Okay, and also you must have at least one authoritative reference. You must list a reference after your text, and you must cite the reference within your text. We call that a parenthetical citation, where you write some information that you've gleaned from your textbook, for instance, and in parentheses you put Anderson et al. 2014 in parentheses, right? That's called a parenthetical citation. So anytime you have a reference, you must cite that within your text. You can't just list a bunch of references. All right, that doesn't work in academia. You must cite the information that you gleaned from those references in your text. You need at least 150 words. So a lot of you are getting comments from me saying minimum substantive word count is 150. All posts and peer responses must be supported by at least one authoritative reference properly cited within the body of your text, like that. And we're using APA guidelines, okay? APA guidelines. You don't have to buy the book. You can Google if you're not sure about it. Plus, the university has provided an APA guide in your success section, student success section, to help you get started on understanding APA. That stands for American Psychological Association. We're using those guidelines to format references and also citations. Some of you still have not, you still missed seeing my announcement about referencing your textbook, Nestor's Microbiology. Go back, it's like the second week or something. I posted an announcement that says this is the way you you format the reference for our textbook, Nestor's Microbiology, 10th edition. Okay, that's the format. All right, don't use any other format. That's the proper format that I posted in that announcement. All right. But remember, your job in discussion is not to say, hey, Joe, you really did a good job on your discussion. I like the way you described Streptococcus aureus and its prevalence in pathogenesis. I also appreciate the way that you incorporated blah, 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 right? Your job is not to evaluate your peers' posts point by point, right? That's my job. right? You're not grading or you're not evaluating your peer's response. You are adding to the discussion. That's why we call it discussion. Your peer, someone in the class, your colleague, posts something about something, and then your task then is to add to what that person has written about that topic, or if you see something, maybe something in that person's post catches your attention, then you can expound on that particular item or that particular point. Someone asked me about this a week or so ago, and I put together a a document for them, starting with the initial post and then the peer response and then their reply back and then my reply also. And it's a good idea. Shows you how discussion is supposed to work. I will post that for all of you to see. It's the two students that are participating in that discussion. They're exceptional students, and their posts are fairly long. You don't have to read everything to get the idea, but you can get the idea of how one person starts a discussion, another person responds by picking up on something that that person said, and then I responded because something that the second student wrote caught my attention. So I did a little bit of research. It doesn't take much. I did like 5 minutes of research, 5 minutes to write my response, and all of 10 minutes, I put together a substantive, um, referenced, supportive, supported response or reply to the second student's post. You can see in that document, I'll post it for you, how discussion is supposed to flow. And again, these students are exceptional. They did, I mean, well above and beyond 150 words. You're only required to do 150 words. And my reply to the second student is maybe 154 words. It's not much. It took me about 10 minutes to research and write. Okay, so that initial post, due no later than Friday, January 30th. All right, there's a microbial diversity assignment or homework and microbial diversity quiz due this week. Both are due no later than Sunday. This is, I think I said January 30 for the Friday. That's actually February 27. And your microbial diversity and microbial diversity homework and quiz are both due no later than Sunday. That's March 1st. Your midterm, again, this is um, midterm. This is the middle of the course. So the midterm exam, it's open now. It closes Sunday. Again, that's March 1st, 11:59 p.m. GCU time. No extensions on the exam. It covers topics from weeks 1 through 4, so including this week as well. It's worth 150 points. And I did post a midterm exam topics review in class resources. I think it's like 22 or 25 items there. In no particular order, I just pulled out some things that I think that you should be familiar with going into the midterm. So check it out. Um, all right. Pay attention as always to due dates on all assignments. Proper credit can only be given for complete and timely submissions. Any questions, issues, or problems, contact me as soon as possible. Message directly 804-229-7993. You can use your Halo message box or inbox. And of course, campus email is good. Links to a video on protists and fungi by the Amoeba Sisters, and then by Professor Dave, viruses, molecular hijackers. Have a great week.